I will be talking about artisanal fishery for squid in the Gulf of California, and that'll be a thread that goes throughout the talk, but I'll talk about some other things as well that I think are really interesting about this squid. This is Humboldt squid. There. So where this talk is going to focus on is the Gulf of California, Sea of Cortez. So as you all know, Steinbeck and Ricketts went there in 1940 on the Western Flyer. And what they saw there, they often described as miraculous and mirage-like. And part of the message I want to convey today is that a lot about the Humboldt squid is also sort of, well, maybe not miraculous, but definitely mirage-like and difficult to discern. But this is a real mirage in the Gulf of California. If you go there, that you'll see this if you're lucky. So these are actually a string of islands that are rising out of the sea like mushrooms. Steinbeck describes this exactly like this. So this is one that I saw in 2011. But the place we're going to be talking about in this talk is the Guaymas Basin. So the Sea of Cortez is made up of a series of deep basins. It's the start of the San Andreas Fault system. The Guaymas Basin is right in the middle. It's flanked by the towns of Santa Rosalia on the Baja Peninsula and Guaymas in Sonora. So this is going to be the main focus of the talk. Different views of Humboldt squid are different things to different people. So this is one popular view of Humboldt squid a few years ago, and I'll play a short video clip.
Weirdest, most beautiful creatures I've ever seen. Just going to release it now.
So Greg, they're not only, they may not be as intelligent as fish, but they're more beautiful. The most beautiful of creatures. This is another view. This is David Attenborough's take on Humboldt squid, also filmed in the Gulf of California.
Thought that the squid flash red and white, not only to confuse their prey, but also to signal to each other when they are about to attack. Humboldt squid numbers are growing rapidly, but they remain mysterious. As fast as they arrived, they disappear back into the deep.
So remember that line about how Humboldt squid disappear as fast as they arrive, because this is going to be a theme that it's going to be a major focus of the talk. Most of that video is true, but it's a little bit exaggerated. This is the view of Humboldt squid in the Gulf of California that would be most relevant to artisanal fisheries. So this is a Humboldt squid being caught by this guy in a panga with a squid jig. They're caught in this manner, one at a time on a handle, and they're thrown into the bottom of the panga. This panga will collect about a ton of squid in the course of a night. So here's one coming back to unload. It's just full of squid in these plastic boxes. They get unloaded at a primitive dock and weighing station. This is all in Santa Rosalia, by the way. And then they are sent to a bunch of factories, local factories where the squid are cleaned and then put back into trucks for shipping to other distributors and mostly to Asia. Actually, they're exported. So the squid fishing began in the late 1970s. You can see here there's really nothing before that. There was a blip in the 1980s. This was mostly Japanese fishermen that were exploring off the peninsula in the Pacific, but also in the mouth and into the Gulf of California. They had their permits revoked around 1983 3 or so, because they were also catching sailfish and a lot of protected fish. So they were poaching basically. So the Mexican government revoked their licenses and hasn't given any squid fishing permits since. And then you can see the fishery really took off in the 1990s. And this initially was, and even throughout the whole course of its history, was primarily artisanal fishery, mostly out of tongas. So these are these small boats. They also catching the squid on hand lines. They also started using retrofitted old derelict shrimp boats, steel boats, out of Guaymas primarily, with maybe 10 or 15 guys that would go out to sea for a week or so, catching lots of squid on hand lines and then taking them back to Guaymas. But it's really still an artisanal fishery. It's a pretty primitive collection method, very selective. You don't catch anything but Humboldt squid for sure. But there were seasonal migrations between Santa Rosalia and Guaymas, which we studied by, well, we talked to the squid fishermen. They told us what the squid did, and then we tagged 2,000 squid all together with little plastic tags for a catch and recovery program, and showed that the squid did indeed go from Santa Rosalia to Guaymas in the fall and then back again the next spring. And some squid would make the complete cycle. The fishery in Santa Rosalia was in the summer. So you can see here that these are the landings in Santa Rosalia, and about 80% of them occur between May and October. So this is the warm season when the Sea of Cortez gets very warm very quickly. So it's a very strong seasonal system there. It's cold in the winter, not much warmer than here actually. And in the summer, it gets very warm indeed. And the productivity, and what I mean by productivity is this type of stuff. Primary productivity produces phytoplankton like diatoms, which provide food for copepods and other zooplankton, and that gets turned into squid food. So these are the things that squid eats. These are myctophid lanternfishes. These are some krill. This is another type of small midwater squid. So the things that squid eat are pretty simple organisms that also grow fast. And the productivity is higher in the cold season off Sonora. They have a lot of winter winds and upwelling, just like off California right now. And in the summer, it's higher off Santa Rosalia. So the squid were following this productivity cycle. So it was a very nice system for them. They could basically eat all year long. And in addition to that, they were spawning within this little closed ecosystem. So these are pelagic eggs that we found at the southern edge of the Guaymas Basin. Blue water divers found them. This is an enormous mass of eggs, the size of a Prius or something. You see the diver in the background. And these were hatchling Humboldt squid that we took by plankton tows up in this area, the northern edge of the Guaymas Basin. And along with, these are called paralarvae because they basically, they're a larva, but they don't metamorphose. So they, larvae of squid are called paralarvae. This is a juvenile Humboldt squid caught in the same area. And in the same area, we have predators of the Humboldt squid, the large ones, which are sperm whales. So this is a group of sperm whales up near this area, near an island called San Pedro Martir. And sperm whales were very common. You could see hundreds of them in this area in a day, just drifting in a boat. So the whole life cycle of the Humboldt squid was happening in this region of the Gulf of California. So they were a pretty obvious organism there. So 1940, Steinbeck and Ricketts went there, and here's their total squid observations. They only recorded 3 species of squid and 2 octopuses of 567 total organisms they found on that trip. So in San Lucas Cove, right near Santa Rosalia, this is the squid fishing capital of Mexico, they hung a light over the side and captured some small squid. And these were identified as some juvenile stage of some loliginid. That's become important in a second, but exact identity couldn't be determined. That was determined by squid expert Barry in a letter to Ricketts. He followed up trying to identify these things. Just north of Santa Rosalia, they hung a nightlight out again and took another small squid. This is in the log from the Sea Cortez only. It's not mentioned in the specimen in Ricketts's field notes or the phyletic catalog or any letters. So I don't know if Steinbeck just thought he remembered this or what, but that's sort of a puzzling one. And then in Guaymas, they went out on Japanese shrimp trawlers, and they took 2 different species of squid. They thought one of these was Loligo opalescens. That's the local squid we have in Monterey Bay, and that was probably misidentified by Ricketts because it doesn't go that far south. And his expert Barry never really answered the question of trying to identify that specimen. So if you dig as deep as you possibly can into their time in the Gulf of California, they really didn't see many squids or octopus for that matter. So why? Here's 4 reasons I've thought about. One was that they just weren't interested, so they didn't pay much attention to squid. Maybe they had too much beer, so they didn't see the squid at night swimming around near the boat. They saw plenty of other things. So there's no way you could spend time in this area in the last 15 years, as I have, and you would not see squid at night from a boat, no matter how much beer you had, because I've tried it every way. They could have misidentified the species. I mentioned that Ricketts misidentified Loligo opalescens, surprisingly. So this is possible. Maybe the squid weren't there. And finally, the squid might have been there, but they were out of view. So as I said, this squid's mysterious. Even Attenborough tells us how they come and disappear quickly. So maybe it's something like that. So lack of attention, I don't think that's probably it. This is Ricketts in the phyletic catalog, and checking our records, it becomes apparent that we failed to get a representative cephalopod picture. Why this should be is hard to say. We covered the region carefully, considering the time limitations, devoted a fair amount of attention to this group, capturing, with one exception, every cephalopod encountered. So they were sensitive to the fact that they didn't see many squid, and this is how they addressed it. So misidentification, this is Ricketts holding a big Humboldt squid in 1936. Before they went to the Sea of Cortez, it was caught in Monterey Bay by sardine purse seiners. He bought like 600 pounds of these things. One of them's in the Monterey Aquarium today in a glass jar, perhaps the same one. This picture was taken by Ralph Buxbaum, who many of you probably know. So Ricketts knew this Humboldt squid, and I don't think it's likely that he would have not recognized it if he had seen it. And even more than that, the small squid that they did capture and identified as lolliginids, this big group of squid, which includes our local squid here, and a really important characteristic of all lolliginids is that they have a cornea. It's a muscular, transparent cornea over the eye. So you can see it sort of here, down here. This is Humboldt squid, Dosidigus. It's a group from the family called Omestrephids, and they don't have any cornea over the eye. The eye is in direct contact with seawater. So it's a pretty obvious visual difference. It's apparent even in a small squid. So I just don't think there's any way Ricketts would have, and Barry would have specifically said these were lolliginids when they, in fact, were not. So I think we can just say they did not, there's no evidence that they saw any Omestrepid squid, which would include Dosidigus. So Santa Rosalia, so did they go there? Well, no. We did not plan to stop at Santa Rosalia. And this is what it looks like today. And maybe the squid were there, maybe not. they didn't stop because it looked from the sea, at least, to be less Mexican than other towns. That's because it was run by a French company. A Mexican town grows out of the ground, but Santa Rosalia looked built. There were industrial works of large size, loading trestles, piles of broken rock. So this is what it looked like, what it looks like today, or a few years ago. And the industrial works are remains of a huge French copper mining operation, which has actually been reopened by a Korean consortium. So they didn't stop there, and this is what it looked like in 1926. So I couldn't find any pictures of 1940, but it probably didn't look much different. And I must say, it doesn't look very appealing as a place to stop, grimy and black and smoky. And I'm reminded of some words from the Cia Cortez when they went to Isla Cayo earlier on in their voyage. We can say it's possible to recognize a shore which is burned, even if it is so far away that the details cannot be seen. And I think this is something that takes us to Steinbeck's concept of warp, and that from the very first sentence of the book, he tells us that this account's going to be warped by his own mind and the way he saw things, and this is the way he remembers the Gulf of California. So it may be that they just didn't go to Santa Rosalia and they didn't want to see what was there because it was ugly and they knew it would be terrible and nothing would be there alive. So that's a psychological factor. But if you look at the history of Santa Rosalia, this is the copper mining production from 1880. It started in 1884 up until the mine closed in 1954. And so they were here in 1940. So the copper mines were still going on. The reserves were kind of running out by 1938, I found. And this is the history of squid fishing. So there was a 40-year gap between the time they were there and the time squid fishing started. So again, it seems that they weren't the only ones not noticing squid. The Mexican fishermen didn't notice squid either, and they're pretty good at reading the sea and knowing what's there, especially if it's something that they can catch commercially. So it turns out that if you look back in the historical records, all the reports of Humboldt squid in the Gulf of California up until 1971 were small. So there were small individuals that were mature. By small, I mean like the one in this guy's hand from the Cal Academy. Dave Powell was on this trip, actually. Some of you must know Dave. And so there were plenty of reports back to 1932 of small squid, mature squid in the Gulf of California, but not a single report of a large one. So they weren't alone in missing these large squid. The first record of a large squid that we could ever find was in 1976. This was an oral history told by a retired shrimp trawler who said he trawled shrimp everywhere from Mazatlan up to the Colorado River mouth from 1946 until 2005 or something like that. So, and he caught one, he remembered exactly where it was, Isla Tortuga off Santa Rosalia in 1976. So we know that the squid were big for the fishery in 2008. This is a rather large one that we captured in the Guaymas Basin. So from 76 to 2008, they were big squid. But this is the same area in 2011. Now this is a mature squid of the same species, and this is what I would call a small one that had been there prior to 1970. So what happened? What went on? And this, I think, is relevant to why Steinbeck and Ricketts may not have seen these smaller squid. So this is a recent history of size of mature squid in the Gulf of California, captured off Santa Rosalia, off Guaymas, as well as from the open basin in the middle. So you can see that before 2010, squid were all big. 60-centimeter mantle length is a huge animal. It weighs 10, 20, 30 kilograms. And then after 2010, they were all of a sudden small. So And there was a little bit of recovery, and then after 2015, they're all small. They've been small ever since, and they're still small. Now, I have also points here from Ecuador, because this squid has a very large range from Chile to Alaska when conditions are favorable, but its core, its center of the range is in equatorial regions, Ecuador and Costa Rica, and the squid never get big there. They're always sort of small. So it turns out that to understand why you have big and small squid, you have to look at what they eat. And so Costa Rica and Ecuador, squid are, these are the mantle lengths of the squid. So in the commercial fishery, let's say, and so they're a mean of 30 centimeters. In Peru and Chile, you have these 30-centimeter ones, even smaller, and then a sort of another bump here, and then you have a large tail of these very large animals. And it turns out that these small ones, which we kind of arbitrarily put at 30 centimeters, live about a half a year, and the large ones live at least 1.5 years to 2 years, and then the medium ones are about 1 year old. And these are all mature, so they mature at different sizes depending on how much food they get. And if you look at the productivity in these areas, this is winter and summer off Peru, and you can see that this is chlorophyll in the water, so it's indicating phytoplankton, which is the base of the food chain for most things in the ocean, and food's available all year round. If you look up here in Costa Rica, it's present in the summer, but not in the winter, so the food's not available year round. So the squid cannot eat a big banquet every day, 365 days of the year in Ecuador or Peru. And so their phenotype, their lifestyle, the characteristic is to mature after 6 months to 1 year, let's say, and they're 30 centimeters long. If they live in Peru and Chile and they're lucky, they can follow these productive areas for 1 year or 2 years and get to be more than 1 meter long. So this is a really interesting lifestyle that's key to understanding the Humboldt squid. So this is the same graph as we saw before. Humboldt squid can only get big in these highly productive upwelling driven ecosystems, like the Humboldt Current off Chile and Peru, and previously, and if conditions are right, in the Gulf of California and even in California. So the important message is that the Humboldt squid in the Gulf of California can take on the tropical phenotype, the new generation, not the adults. The adults don't shrink, but their offspring take on the tropical phenotype. And this is a case of strong El Nino. So these vertical bars here are strong El Ninos in 29 and 2010. So an El Nino, it seems, results in the squid of the next generation maturing when they're small. And the same thing happened in 97 and 98. So 3 different El Ninos now we have data on this, and the same thing we've seen each time. So El Nino is a super important signal to these animals. So why is that? So I'm going to be out of time, so I'm going to skip what an El Nino really is, but what an El Nino does is drive warm water from the surface to very great depths. And it happens every 5-10 years. It's unpredictable, but they know now when they're coming, and there's none coming in the near future. But this is a warm water anomaly. So the water's 4 degrees warmer at 200 meters deep than it should be. This is during the 97, 98 El Nino. It's very strong. And this water that's coming back across the equator with the El Nino is also very low in nutrients. So that means if you have upwelling, there's no nutrients in the water that gets upwelled, so everything starves basically. So what's so bad about El Nino? Well, if you're a squid, it means warm, nutrient-poor water at depth makes futile upwelling. There's no food in the upwelled water. And so the phytoplankton blooms that support zooplankton and the things that squid eat just don't happen. And so that these effects can ripple through many higher trophic levels in these upwelling dependent ecosystems from squid to marine mammals. So it's a very big deal for the animals that live in these upwelling areas. And so it turns out that this temperature sensitivity is reflected, I think, in the way squid behave. So temperature and nutrition are linked to foraging behavior of the Humboldt squid. So this is an example of Humboldt squid at 200 meters depth eating krill. And at 200 to 300 meters depth, the water's cold, there's not much oxygen, and the food is somewhat lethargic. So the squid can just sort of gobble it up. At nighttime, the squid always, every place we've tagged them with these satellite tags, hunt in the upper 50, 75 meters of the water column. So it's much warmer and the food's much faster. So it turns out, though, that if you look at the behavior of the squid with these tags, the squid never stays above 22 degrees for very long. If it enters water 22 degrees or more, it dives back down to cooler depths after only a few minutes. So it's trying to go in that warm water to eat. It goes into water up to 32 degrees, but only for a minute or 30 seconds, let's say, and then it gets something if it's lucky and goes back down to cool off. So this leads to the idea that if the temperature in the upper 50 meters, 75 meters of the water column is higher than 22 degrees C, then the squid can't enter that region to eat because they're thermally prohibited, basically. It's a hostile area to them, it's too warm. And so this means that if their food is up in that zone at nighttime, then they have lost access to their really important part of their calorie intake. So it's highly unfavorable, stressful, and this is why probably El Nino triggers them to take on that small phenotype. And this is just now a history since 2005 of temperature in the water column off Santa Rosalia. And this is color coded from 30 degrees down to 15, but this is a 22 degree depth contour, basically. So at this depth, it's 22 degrees. Here it's back before 2010, it was 30 meters. 2009 and 10, it goes down to 60 meters. It recovers a little bit, and then it goes down in 2014 and 15 to 75 meters, and it's slowly coming back. So during most of the time from 2009 to 2020, this 22 degree C thermocline or temperature depth was more than 50 meters. So it was unfavorable for squid. So we think that's the reason why they got small during El Nino, following El Nino, and then they remained small for so long. And now things are looking like they may be recovering, and it's going to take several years, I think, before we can tell what's going to happen again. This is commercial landings now. So the depth, this is the size of squid versus this depth at 22 degrees C. So between 2010 and 20, it's red because it's below 50 meters. So this is the hostile for squid area, and the squid are all small. The commercial fishery for squid follows exactly the same trajectory. So after 97, 98 El Nino, it crashed, but it recovered again. And then after 2010, it crashed, started to recover, and then 2015 just put an end to it. And it's not because there's no squid here. The squid are present, but they tend to be much further offshore than the large ones. They take up a different pelagic blue water habitat than the large ones. That's another characteristic of the phenotype, in addition to being small. And the smallness means that commercial fishermen can't catch them. So the fishermen have just given up trying here, because when you have a 60 centimeter squid, these are the types of squid jigs you use, like you saw in those videos. When you have a 20 centimeter squid, and this would be what it would look like compared to the 60, you can't catch them with these things. It's impossible. So you can't even catch them with squid jigs of the small type that people use in California. So we had to start using these little 8-centimeter jigs we get from Yozuri company in Japan to catch these little squid. And you can catch them with those, but it's very tricky. It's kind of like trout fishing, and you certainly couldn't catch a ton of them at night to make a living for your family. So the fishermen have given up. I'll stop there, and just to say that in 1940, when Steinbeck and Ricketts were in the Gulf, we don't know what the conditions were, but if we go back in time, looking at these subsurface temperature data, the earliest ones I could find were 1955 to 1960, and the conditions were definitely unfavorable. This 22-degree depth barrier, depth thing is well below 50 meters. And this is 1970s where the depth barrier gets good again. And here's 1976 when the first big squid was found. From then on, conditions must have been good in the Gulf of California because there were big squid, and 2010, they became unfavorable. And as I say, they're recovering now, and it remains to be seen what comes back. So who knows? So that's all for me. So thank you.
Is my slide up for everybody? Yes? Okay, I'll start the show. Okay, I hope everybody can see that. That's sort of my title of the talk today, but not of all of the papers we've written. We're looking at a whole different group of organisms, sharks and rays in the Sea of Cortez, and we did a survey for 2 full years, 1998 through 1999. And these are organisms that were not neanderthal and not those that the Western Flyer expedition surveyed. So that's what I'm going to be talking about today. There were a lot of researchers involved with this 2-year survey, Bob Huter and John Kaminsky from Mote Marine Lab, myself and 2 graduate students at the time, now PhDs, Joe Pizarro and Wade Smith, and several Mexican scientists, Fernando, Leonardo, and Carlos. All of 3 now have PhDs and are involved with institutional work in the Gulf of California. We had funding from a total of 12 different institutions, sources, including our own laboratories, that helped fund this project, including the David Packard Foundation. The Gulf of California is also known as the Sea of Cortez, as Gilly mentioned. It's 1,100 kilometers long, 160 kilometers wide, 3,000 kilometers of coastline in all, very high biodiversity. And here I'm talking about fishes and invertebrates, and high fisheries yield. But the data on the fisheries catches is not as extensive as what Gilly talked about. The largest numbers and biomass of sharks and rays that landed in Mexico were in Baja California, in the Gulf of California. So there's a statistic, and that's the area we surveyed. Elasmobranchs, or sharks and rays, grow slowly, have a late sexual maturity, have low litter sizes and or egg production, annual or sometimes biannual reproductive cycles, and they're at a very high trophic level, pretty much like squid are, but among the fishes, they're definitely at the top. They're highly vulnerable to overfishing. The project objectives were at least 6: to locate all artisanal fishing camps along the whole coast of the Gulf of California for a 2-year period. This had not been done before. We convinced the funders to do it because there had been large entrances of fisheries from other countries, China and others, taking large sharks, and we wanted to make sure we had an idea what there was there. So we collected camp characteristics and catch and effort data for all the camps we encountered. We determined catch composition and shark ray catch per unit of effort, CPUE, for camps, most of which were targeting sharks and rays, but occasionally got bony fishes as well. And we were looking for shark nursery areas and other things that would help us understand the shark and ray populations in the Gulf of California. We had to learn to identify the shark and ray species at risk. This is not just 1 or 2 or 3 squid, like Gilly talked about, but there are a lot of species that we had to deal with in all stages of life. And we wanted to produce recommendations for conservation. So there is the Gulf of California we surveyed. We did it by vehicle, not by boat, like the Western Flyer did it. And we would go to fishing camps where they would bring in their catches. There were 2 camps on the left, on the west, Baja California Norte in blue, Baja California Sur, 83 camps in green. The Moss Landing folks covered the top blue ones. The people from UABCS covered the 83 camps below with some help from the other groups. And then on the east side of the Gulf, the west side of Mexico, both Sonora and Sinaloa, there were 19 camps and 28 camps. The total, I can't read it because people are in the way, but I think it's 147. So that's how many camps we found with all that survey we did. It was a lot of work. This is a typical picture of what goes on. There are students, this one's from Mexico, from UABCS, and from here, and the pangas that come in and bring in the sharks that they've caught, and we would work them up to produce information that allowed us to characterize the fishery for all these species. This will bore everybody, but basically we took data on general camp characteristics, boat and gear types, target species, and economic data. Those were our camp survey data sheets. And we had a panga per boat survey data sheet, which talked about the size of the boat, the fishing method and collection, the catch composition, and the individual shark ray data that we found from them. We have 5 papers that are published, and later on when this slideshow is available in tape form, if you want any of these papers, please let me know. I can send you PDF reprints of any and all of them that you'd be interested in. I'm not going to go through the titles, but they're all by the different groups that studied the 4 locations in the Gulf of California. The project results, fishing camps ranged from remote primitive camps to some of them large towns and cities. And also they used gill nets primarily, but sometimes long lines and hand lines with hooks and bait were used. There were 147 camps, as I mentioned, comprised of 4,000 active pangas, and as many as 5,500 at one stage of the study were found. Most of them target sharks and rays. We surveyed 455 sampling days, all of us, the groups. We saw 165,937 sharks and rays. 9% were measured by researchers after being identified, of course. Now, you're not going to be able to read all this, but there were 30 shark species on the left. That column lists them, 24 ray and skate species on the right column. And we listed those to figure out what species of sharks, rays, and skates were found in this fishery. And basically, as you can tell, it's quite a high biodiversity number of species of sharks and rays or elasmobranchs in the Gulf of California. Pie diagrams for 4 locations. This is Baja California Norte, comprised mainly of sharks in the genus Mustelus, rays in the genus Rhinobatus, and bony fish dominated by croakers, cyanids. We surveyed 7,435 specimens of those in that location. In Sonora, across the bay, we primarily saw sharks, the blue, and rays in green, and surveyed 157,787 sharks and rays. In Baja California Sur, there were more bony fishes. Scambra morse is one of them, but quite a few angel sharks, blue sharks, hammerhead sharks, and mobulids, with a total of 1,281 sampled. And then finally in Sinaloa, across the bay to the east, the hammerheads and bonnetheads dominated. I have to go back, sorry. Dominated the catch, and some of the rays, Daisyatus, Rhinoptera, and Rhinobatus were there as well. And the bony fish here are in red. We surveyed 3,717 specimens there. Typical picture of one of the fishermen. Everybody's dressed warmly because it's hot down there, but only 22 billfishes were found. So this indicates they were fishing offshore farther than Ricketts and Steinbeck surveyed, but not as far off as they would get billfish. They were primarily surveying coastal sharks and rays. Here's a list of all the species. The point is relative frequency in the catch. Some of these have high abundances with 3 asterisks, some of them intermediate with 2, and some with low ones with 1 asterisk. I don't expect you to understand that, but I want to give you the idea of the details we went to characterize the fishery so it could be monitored in the future. One point, and underlined on the right side, is these are the species that were caught that had juveniles. In other words, they're sacrificing young of the year sharks and rays, which is not a good thing to do in many fisheries. So we were kind of curious about that, and we tried to document that. Along those lines, we also kept track of which ones had pregnant females documented. There were 28 species that had those, and those are noted also by the symbols on the left. I'll show you some more details in a minute. Right now, as a matter of fact, neonate or newly born or young of the year scalloped hammerheads were taken in those locations, and these are females, and the big ones are noted as a larger symbol. The smaller ones are noted as smaller symbols. So as you can see, they were taken in all 4 locations that we looked at the scalloped hammerhead. Several of them were pregnant females or young born babies, neonates. The other hammerhead, the smooth hammerhead, didn't get quite as large, but the females were primarily in Sonora and down in Baja California Sur, and the large ones primarily in the middle of the Gulf. Okay, and then the third species that we noticed that had females and had neonates associated with them, newborns, were the black-tipped sharks shown in this picture here, primarily in the Gulf. 2 pictures of pregnant females, the black-tipped shark in the upper left-hand corner, and the banded guitarfish, which are live bears in the Gulf of California, showing some of the offspring that they had. We took a lot of care to detail that. So the general results, I've got to back up again. The general results are the species diversity in the elasmobranch catch is very high. 30 species of sharks, 22 species of rays, and 2 species of skates, but they weren't as common because they're mostly deep water. There was a shift seasonally throughout the Gulf. Latitudinally, they were similar, i.e. Baja California Norte and Sonora were similar, and Baja California Sur and Sinaloa were similar in catch patterns. Juvenile sharks and rays were common in the catch, both as neonates and older juveniles. And unfortunately, pregnant females were also common in the catch, and that was primarily in the summer. Results continued. The peak of the large sharks, they call them tiburones in Spanish, is summer, but there has been a lot less targeting of large sharks in recent years, primarily, we think, because they're not as available. Rays, called rayas or mantarrayas, and small sharks, cazones, are an important component of the catch much of the year, especially in the spring and summer, as mentioned earlier, with rays becoming more important later in recent years, mainly because the large sharks had primarily disappeared. The utilization of the catch by the fishermen is high, and it's mostly local. They don't ship them away. They use meat, fins, and other products harvested from all elasmobranch catch. Many of these camps don't have refrigeration. Most of them don't. Some of them have ice, but they normally use salt to preserve them. And then they would ship them out by truck to the different locations where they would be consumed after soaking. Something I've got to go back again. Juvenile pregnant sharks are common. This is the second result. The juvenile sharks are common in the catch, both as neonates and older juveniles. And as I mentioned, pregnant sharks are common, with the large sharks and rays in the summer. I think I've gone through this before. Okay. The size and abundance of sharks down here, number 8, appears to be declining in the Gulf. And it looks like many locations have local shark stock depletions, mainly due to the evidence we saw for them shifting from one place to another. Shark products in Mexico are used for fillets on the upper left, on the right, the carcass, on the lower left, shark fins, which is highly profitable, and on the lower right, cartilage. Shark and ray fish, artisanal fishing still occurs in the Gulf of California. Because of our pioneering surveys, they're now being monitored by the Mexican fishery biologists and enforcement authorities. So we're getting additional data and hope that that'll help with management. The lessons are overfishing of sharks and rays can still occur even though there's high abundance, there was high abundance and biodiversity. They still can occur. Even with seemingly low-level, non-industrialized methods of fishing for sharks and rays, overfishing can still occur. Elasmobranchs, due to their life histories, as I mentioned early on, are highly vulnerable to overfishing. It's imperative that Mexico implement fishery management plans for sharks and rays as soon as possible. And that's the end of my brief overview of our large survey. Thanks for your attention.